Good day, everyone. I'm Dr. Anna Peltseva, a new faculty member at University of Louisiana at Lafayette School of Geosciences. I'm pleased to share with you the work done in New York City as part of my doctorate research at CUNY Graduate Center and Brooklyn College. New York City, the largest and most densely populated city in the country, with the years of industrialization that have left soil concentration of lead and arsenic in many parts of the city. In my research, I visualized the extent of lead contamination in New York City urban gardens, assessed how lead and arsenic affect plant metal levels, and developed a new method to assess the degree of human risk based on the nature of lead in soils. In this talk, I will focus on benefits of urban gardening and common contaminants, soil contamination in the city urban gardens, variability of bioaccessible lead, case studies in urban and suburban gardens, rapid screening of bioaccessible lead in urban soils using novel methods, mitigation strategies and emerging soil amendments. And I will conclude with perspectives for future research. Urban agriculture actively engages 800 million people worldwide, and urban cropland covers more than 5% global cropland area. Gardening brings many benefits to urban residents, such as nutritious food, grass rubble reduction, physical and mental health benefits, decreased crime, a deep connection to agriculture and urban nature, and even promotion of biodiversity. During the pandemic, due to COVID-19, gardening also has contributed to food security. Urban soils are often contaminated with organic and inorganic compounds. The former include primarily dioxins, furans, pesticides, and hydrocarbons. The latter include lead, arsenic, barium, cadmium, copper, zinc, nickel, and mercury. While all contaminants should be monitored for impact on human health, alongside emerging substances such as microplastics, we focused on lead and arsenic, which are by far the prevailing toxicants in urban soils with the highest rates of toxicity through ingestion. Lead is relatively immobile, while arsenic very slowly leaches through soils. In practical terms, this means lead and arsenic compounds contaminating soils in the late 1800s persist today. As urban vacant lots and former orchards are being converted to residential areas, the accumulated soil levels of lead and arsenic pose a threat to human health when gardeners grow crops in this soil. New York City is the second highest after Quezon City in the Philippines in terms of lead and cadmium contamination, and after Dublin, Ireland in terms of arsenic. Using geospatial tools, I interpolated the distribution of lead in New York City gardens and found the highest concentrations were located in the former industrial areas of northern Brooklyn, where there had been a lot of smelters, manufacturing plants, coal yards, and incinerators. Total lead concentration ranged from 3 to 45,000 milligram per kilogram. The contamination level decreased, although toward the suburbs, which is also evident in many cities across the United States. We can see here the distribution of soil samples around the city, most of them concentrated in Brooklyn, in the northern Brooklyn. We had over 2,000 samples for this data set. Contaminants can get to a human body through consuming contaminated crops, drinking contaminated groundwater, but mainly through direct ingestion and house dust. It's been well established that bioavailable lead absorbed by internal organs potentially causes irreversible fetal brain damage, hearing problems, and learning disabilities. However, we found that not all of the total lead poses the same threat to human health. We use bioaccessibility of lead assessed in lab conditions as proxy for bioavailable lead, which is typically tested on animals. We found that bioaccessibility of lead in 49 urban soil samples from New York City varied among samples and with the pH of the extractant solution used. We used the 1340 method developed by EPA, and the extractant solution was 0.4 molar glycine at pH 1.5 or pH 2.5. Lead at pH 1.5 was extracted from 14 to 86%. At pH 2.5, it ranged between 14 to 73%. With higher phosphate and organic content, lead bioaccessibility was lower at pH 2.5. As also seen on the slide, there is no relationship between total lead and bioaccessibility of lead. Some samples with a low concentration of total lead that may have high bioaccessibility of lead. These findings highlight the heterogeneous nature of urban soils and the need for using bioaccessibility rather than total concentrations for better assessment of human exposure and risk. When calculating relative bioavailability of lead, we found that most of gardens measured were at low risk. In general, it's recognized that soil bioavailability and bioaccessibility are controlled by the speciation of contaminants in different soil phases. However, there is a great variation in the bioavailability of metals in soil particles. Lead, arsenic, and other toxic metals speciation is dependent not only on the source and chemistry of the contaminants, but also on the mineralogical and biophysiochemical properties of the soils. There is a particular interest in the phosphate content of the soil, as there are complex interactions between phosphate and lead, and phosphate additions can be used to reduce lead hazard. There is a clear need for further research on the factors regulating bioaccessibility in urban soils and for the development of rapid, inexpensive methods for assessing bioaccessibility. The steps will facilitate the development of guidelines for lead exposure based on actual bioaccessible lead rather than total lead. The current guideline is based on the assumption that bioavailable lead is 60% of total. Back in 2014, the New York Post published 2 articles on toxic soils and toxic vegetables grown in Sterling Community Garden in Brooklyn. The stories were based on the data collected by Professor McBride and others from Cornell University, and used phrases such as disturbing new state data, the numbers are startling, and this is insane. However, our research showed that only a small fenced area was contaminated and not the entire garden. Here's the map of our measurements, and only this area under the peach trees that was fenced had high concentrations of lead and arsenic. We established here experimental plots in this fenced area and planted tomatoes, eggplants, kale, onions, and amended soil with bone meal, compost, sulfur to activate phosphorus-enriched soils for metal immobilization. Our analysis showed that the dominant risk for metal exposure at this site is soil and dust ingestion. Consumption of vegetables calculated based on measured geometric mean of plant contaminant concentration, vegetable consumption rates, and assuming 100% bioaccessibility from the amended plots would also exceed also exceed the health safety limit of 3 microgram per day. To reduce ingestion rate to less than 3 microgram per day to ensure children's safety from incidental soil ingestion, soil should have 80 to 90 milligram per kilogram of total concentration of lead. This is consistent with Dr. Milke's research in New Orleans, who stated that a safe soil level for most children is about 80 milligram per kilogram. Soil and dust lead ingestion rates from this study were also compared to a plot established in the study area and published in another paper by Eggendorf in 2018, with constructed clean soil, which had exposure values with much lower than FDA threshold of 3 microgram per day. Estimated daily lead or arsenic exposure were very low, since soil concentrations in these plots were 20 milligram per kilogram of lead and 1 milligram per kilogram for arsenic. Suburbs aren't exempt from contamination problem. While lead was popular in cities as versatile material for paint, pipes, fuels, and insecticides, lead and arsenic were commonly found in pesticides used in suburban apple and peach orchards and potato farms to fight gypsy moth. Generally, root vegetables are more susceptible to metal contamination, followed by leafy greens and then fruits. Metals adhere on root vegetables. Peeling help reduction of plant tissue concentrations of metals, except for carrots, that uptake lead through their cores. Leafy greens get contaminated by splashes and dust deposition from surrounding areas that cannot be completely washed away. Fruit vegetables, for the most part, had very negligible concentration of metals in their tissues because they have very strong physical barriers inside of them, and also the fruits were very far away from the surface and didn't get the splashes. We found variability among different groups of plants. It's often believed that plants are contaminated through root uptake, which we found isn't always the case. Moreover, significant levels of lead and arsenic trapped in soil particles on plant tissues of root vegetables and leafy greens are impossible to wash off. Fruit vegetables grown in soils mildly contaminated with lead and arsenic were still safe for consumption. As indicated earlier, there is a need for simple tools and methods to perform diagnostic screening to detect hotspots of contamination and associated risks and prevention potential human exposure. A new approach for low-cost and rapid assessment of soil lead bioaccessibility was proposed using a portable X-ray fluorescence analyzer. The procedure includes the following steps: air drying of soil samples, extraction with the soil extraction ratio of 1 to 10 for 1 hour at room temperature, filtration of the extract, and direct measurement of lead in the liquid extract in cups of portable XRF. The extractant can be nitric acid or glycine solution that we both saw working well. The results show that XRF was able to measure lead directly in these liquid samples, and XRF and ICP mass measurements of the same extractants were highly correlated, highlighting the potential of using pXRF for liquid analysis. Scientists from Columbia University developed a soil bioaccessibility kit to measure hazardous levels of lead in soils. The procedure for the kit is derived from the in vitro bioaccessibility method of Jackson and Bright in 2007 and EPA method 1340, but uses a higher soil to solution ratio. The field procedure extract solution can be measured by both XRF and ICP-MS for quantitative results. The use of visible and near-infrared reflectance spectrum-based instruments can potentially reduce the time and cost of soil quality assessment. Visible NIR with the wavelengths ranging between 350 and 2500 nanometers allow the detection of soil components with the spectrally active elements. The results of our analysis indicated that soil organic matter and iron have important roles in the estimation of lead concentrations from the visible NIR spectra of the soil samples from New York City. The strong correlation between lead and zinc concentrations suggested a common source from both leaded gasoline emissions and lead-based paint. The close correlation between the concentration of lead and the organic content is the major mechanism. Given the strong adsorption ability of organic compounds, we obtained a successful model for estimation of bioaccessible lead. We found a close correlation with total iron underlying the presence of other sources of contamination from visible NIR spectra. Further study will investigate which variable, organic content, iron, or maybe something else, predicts best lead bioaccessible concentrations. Our research showed that suburban and not just urban concentrations of lead found in soils is not just the suburban, is not just urban, but also suburban problem. And vegetables produced have metals at levels that above healthy or safety standards in some varieties like carrots or radishes, and also leafy greens. Kale and lettuce are found to have excessive concentration of metals in their tissues from splashes and dust deposition. We found that plant type was the most important predictor of contamination rather than amendment type. After a series of trials to reduce contamination and remediate the soil with phosphate-bearing amendments, we concluded that adding compost significantly improved the soil quality, plant health, and yield, while decreasing soil and vegetable contamination levels. Compost is the great way to recycle organic matter, reduce food and yard waste, sequester carbon, but also, depending on the source, it may have some contaminants. Using tree leaves may have a high lead levels, especially near the street. Leaves are often gathered at the curb where street dust high in lead is also collected. Manure is usually a low metal organic amendment, but near cities with smelters, manures are heavily contaminated and should not be used. Swine and poultry feeding programs use high levels of copper and zinc to increase feed use efficiency that accumulate in the compost. This is important to remember when choosing a vendor for compost. Amendments have variable effectiveness in reducing the bioavailability of lead and arsenic and may not always work. Organic matter on the other hand, dilutes metal concentration and binds them into stable organo-mineral forms. On this slide, we can see a whole range of organic waste that can benefit soil health and lower toxicity of soils, all the way from fruit peels to tea leaves, sawdust, nuts, walnuts, coconut shells, seaweed, coffee grounds, and so on. Other organic waste products that gain their momentum are biosolids and biochar. Biosolids are treated sewage sludge that promote greater plant biomass, nutrients, sequester carbon, immobilize harmful metals, but may also contain some pollutants. So it's important to know where it is coming from. Biochar is a charcoal combusted at high temperatures and low oxygen, sequestering carbon. It improves water quality, reduces soil emissions of greenhouse gases, provides nutrients, and also can bind heavy metals. Lead and arsenic contamination is prevailing in urban soils and as a major constraint on urban agriculture and gardening. As city dwellers increasingly embrace urban gardening for fresh vegetables and personal pleasure, it is essential that we are able to identify highly contaminated areas as priorities for remediation. We must also educate the public regarding soil health and enable them to test and remediate soil without lab equipment. And the best remediation practice is using compost to dilute and bind metals with organic matter and using raised beds filled with clean soil. We should focus on promoting these public resources. For example, the New York City Clean Soil Bank, which is the glacier sediment extracted from the deep horizons in the city during the construction. These sources can be found across the country and ultimately the world and used for upcycling material and remediation of contaminated soils. For future research activities, I highly encourage further research in vivo studies on urban soil amended with phosphates. Synchrotron analysis of lead and arsenic speciation for understanding the mechanisms that trigger lead and arsenic bioavailability in urban soils. Studies on recontamination of remediated soils and vegetable surfaces. Modeling sustainable urban food production with related ecosystem services. And a quantification of carbon sequestration in urban agriculture. How much we actually store in soil when we do add all this amazing organic waste. The current environmental issues associated with the impact of climate change, global warming, and extreme events on soil contamination and metal mobility in soil requires special attention. In the case of dry seasons, the addition of phosphate fertilizers, compost, and soil amendments may not form stable and thus less bioavailable forms of metals due to the lack of water content required for chemical reactions to occur. Crop concentrations of metals tend to increase when soil temperatures are higher. Yet it is not clear if this is due to accelerated evapotranspiration, more rapid organic matter breakdown in soil, faster release from soil particles and diffusion to roots, or some combination of several factors. Thus, practices for soil erosion prevention are vital to maintain soil health and sequestration of contaminants in urban soils. I would like to thank all researchers, contributors, friends, and students who helped to accomplish this big work and our funding agencies for supporting us. If you have any questions for me, please do not hesitate to reach me at anya@urbansoils.org. Thank you so much for being a part of this conference and listening to my presentation.